The electron microscope, which was applied to the study of biological tissues in the 1940s, permits magnifications 2 to 3 orders greater than is possible by light microscopy. As a result of observations of tissues with this greater magnification, a number of new structures were seen. These are illustrated in this plasma cell from Bloom and Fawcett's classical textbook of histology. The new structures are the endoplasmic reticulum, first seen by Porter, Thorpe and Fulham, the nuclear pores by Callan and Tomlin, and the unit membrane by Robertson. This well-known diagram by Brachet of the generalized cell is one of 2 which appeared in the Scientific American and have been widely reproduced in textbooks. In it, the following features can be seen. Within the cytoplasm there is a Golgi body and an extensive endoplasmic reticulum which is connected to the extracellular space and to the nucleus. Nuclear rotation is illustrated by this cell from a culture of rat hearts speeded up 48 times. Let us consider the endoplasmic reticulum. The first question that arises is, how is it possible for the intracellular movements which we have seen in living cells under relatively low power light microscopy and, according to modern textbooks, is attached to the cell membrane and to the nuclear membrane? All intracellular structures are moving continuously and at different speeds. If we were to stop this movement and draw the generalized cell, it would look like this diagram. The membranes around the cell, the nucleus, the mitochondria, and probably the nucleolus are of single thickness and are imperforate. There is no endoplasmic reticulum or Golgi apparatus.
From DNA to messenger RNA, and we call that process transcription, and this occurs in the nucleus. And then that messenger RNA makes its way outside of the nucleus, and it attaches to a ribosome, and then it is translated into a protein. And so you could say that this part right over here, this is happening as this is being facilitated by a ribosome, or it's happening at a ribosome. Outer nuclear membrane, you could continue this outer nuclear membrane, and you get into these kind of flaps and folds and bulges, and this right over here is considered a separate organelle. So you get this thing that looks like this, and I'll just do it the best that I can draw it. And this right over here is called the endoplasmic reticulum. So this right here is endoplasmic reticulum, which I've always thought would be a good name for a band. And the endoplasmic reticulum is key for starting to produce and then later on package proteins that are either embedded in the cellular membrane or used outside of the cell itself. So how does that happen? Well, the endoplasmic reticulum really has 2 regions. It has the rough endoplasmic reticulum, and the rough endoplasmic reticulum has a bunch of ribosomes. So that's a free ribosome right over here. This is an attached ribosome. This is a ribosome. These are ribosomes that are attached to the membrane of the endoplasmic reticulum. So this region where you have attached ribosomes right over here, that is the rough endoplasmic reticulum. I'll call it the rough ER for short, perhaps an even better name for a band. And then there's another region which is the smooth endoplasmic reticulum. And the role that this plays in protein synthesis, or at least getting proteins ready for the outside of the cell, is you can have messenger RNA, right? Let me do that in that lighter green color. You can have messenger RNA find one of these ribosomes associated with the rough endoplasmic reticulum, and as the protein is translated, it won't be translated inside the cytosol, it'll be translated on the other side of the rough endoplasmic reticulum, or you could say on the inside of it, on the lumen, or in the lumen of the rough endoplasmic the rough endoplasmic reticulum. Let me draw that a little bit better. So let's say that this right over here, that right over here is the membrane of the endoplasmic reticulum. And then as a protein or as an mRNA is being translated into protein, the ribosome can attach and let's say that this right over here is the mRNA that is being translated. Let's say it's going in that direction right over here. Here is the membrane of the ER. So ER membrane. This right over here, and actually the way I've drawn it right over here, this is just one bilipid layer. So I could draw it like this. I could do it like this. And this bilipid layer is continuous. It's continuous with the outer nuclear membrane. So let me just make it like that so you get the picture. And then at some point in the translation process, the protein can be spit out on the inside. On the inside, as it's being translated, it can be spit out on the inside of the endoplasmic reticulum. So this is the lumen. This is the ER lumen right over here. So we're inside the endoplasmic reticulum here. Here we're outside. Here we're outside in the cytosol. In the cytosol, outside in the cytosol. So that way you get the protein now inside the ER, inside the endoplasmic reticulum, and it can travel through it. And at some point it can bud off. So let's say, imagine the protein is right over here. And the smooth endoplasmic reticulum has many functions, and I won't get into all of the depth of how it's involved. But at some point, that protein can bud off. And so let me draw a budding off protein. So let's say this is the membrane of the endoplasmic reticulum, and a protein, let's say, ends up right over here, and then it can bud out. So it could go from that to, let me do that same color. It can go from that to that, to that. I think you see where this is going. To that, and then to that. And then it could go to something like this. Now it has budded out. And when you have a protein, or really you have anything that's being transported around a cell with its own little mini membrane, we call this a vesicle. So now it'll bundle up, and now it is a vesicle. Now this vesicle can then, let me draw some of these vesicles holding some proteins. So let me draw that. Can then go to the Golgi apparatus. The Golgi apparatus, which I'll draw in blue right over here as best as I can. So the Golgi apparatus, this is not, obviously there could be better drawings of something like this. And then they can essentially do the reverse process, and they can attach themselves to the Golgi, oftentimes the Golgi body, named after Mr. Golgi, who discovered this. And then the proteins, once they get into the inside of the Golgi body, then they essentially go into a maturation process so that they're ready for transport outside of the cell, or maybe to be embedded into the cellular membrane. So this right over here is the Golgi body, or a Golgi apparatus. Golgi apparatus. Or Golgi body. And then once they're done with that process, then this is kind of the fully manufactured protein ready to be used. And actually, yeah, maybe I'll make it in a slightly different, well, I'll just use that same color. This is the fully manufactured protein. And now it can transport to the cell membrane, and that protein can either be transported outside of the cell, or it can be embedded within the membrane itself.